Okay, shall I start? ओपन मोबाइल में ओपन करो, जो स्टडी मटेरियल मॉर्निंग में भेजा था उसको ओपन करो. Is this visible? Regulation of gene expression.
Yes, sir.
Yes. Okay. So basically, what do you mean by gene expression? So it is the process by which information encoded in a gene is converted into a protein. And it determines this protein, it determines the organism's characteristics and function. So essentially, your function or your phenotype is the protein which is expressed by that gene. And it happens through a process known as transcription, which then goes to translation. Fine. So next slide, go to the next slide. DNA, transcription, RNA, the protein. So exactly this is the process which takes place. Go to the next slide now. So, and the central dogma, you know, DNA to RNA, RNA to protein, and sometimes RNA can come back and form DNA through a process known as reverse transcriptase. And DNA replicates itself. This is the central dogma. And so, next slide. There are essentially 7 processes that affect the steady state concentration of a protein. So, if your phenotype is a protein, the regulation of that protein, how much more of that protein will be there or how less of that protein will be there? That is the question. How much or how less? That is controlled by regulation. Like a fan, you have a regulator. How much faster will it rotate or how slower it will rotate? So this is exactly what you mean by gene expression regulation. How much more, how less? This is how it goes. So there are 7 processes which affect the steady state. Steady state means the same concentration of protein. So there are 7 processes which increase or decrease the steady state. It's going at a X concentration. Suddenly, because of some particular impulse or something, it's gone to 2X concentration, or it's become X by 2. So the first place where we do is the transcription initiation, first step. Okay, then you have the primary transcript which is forming, and then the second step which you have is the post-transcriptional processing, splicing, capping. Then the third thing which comes is the RNA stability. The next thing is the translational regulation. So there is a translational regulation also which takes place, which is controlling how much translation will take place or how less translation will take place. So translation will make the protein. So this is how regulation is done. Then the protein is made, but then the protein needs to be modified. Ubiquitinylation, the protein acetylation, methylation, they make the protein active. Phosphorylation, they make the protein active or inactive. So depending on this, the final expression of the protein, functional expression of the protein is affected. So that the next step is you have seen the protein modification, protein degradation. Proteins are tagged with a moiety known as ubiquitin, and once it's ubiquitinated, it is targeted for destruction, ubiquitinylation. So if the ubiquitinylation it is very high, then the proteins get destroyed very fast.
স্যার কৃতিকারা জয়েন হতে পারছে না।
না স্যার, জয়েন হয়ে গেছি।
ঠিক আছে।
And the one more thing is protein transport. So the protein, after it is produced in the cytoplasm, it needs to be transported to the cell membrane or to the Golgi body or to the ER or to different organelles. So if you stop that part, final expression of that protein will also be affected. So basically, these are the 7 steps of regulation. Go to the next slide. So in the past, the focus has been always on understanding transcription initiation. That is, what are the transcription factors which are binding to the promoter and causing the transcription? The transcription factor, the DNA binding element, what are the elements of the promoter? These are all the things which were in the mind, final expression of the protein. But nowadays, there is lots of stress that post-transcriptional, post and transcriptional, translational regulation, protein and protein DNA contact. So this gene regulation essentially happens through protein contact. Two proteins are interacting or protein DNA transcription factor binding to the DNA. So this is how essentially what is the current trend in gene regulation. So two things you have to understand. The next slide. One is housekeeping gene and the second is regulated gene. So a housekeeping gene is that gene which is throughout, it is essential for housekeeping activities. Hotel mein, you have a housekeeping activity. That is, what is the housekeeping activity? Room maintenance, bistar ka chadar clean karna, bathroom clean karna, all the saaf karna, fan, lights, sab connection theek hai. These are housekeeping. Every day ye kaam karna padta hai, bartan dhona. So these are all housekeeping activities. Special occasion nahi hai. And then when this Narendra Modi comes, suddenly the hotel is decorated with lights, flowers, welcome banners and everything. That is regulated. Because of that particular impulse of this Narendra Modi coming, maybe the hotel name will also be changed to Narendra Modi Hotel. He's a narcissistic personality. Yeah, so whatever be it, but this is regulation. It can be inducible, बढ़ जाता है. It can be repressible, घटा देता है from housekeeping activity. Now, genes and regulatory. What are the regulatory elements? Next slide, mein dekho. First are the structural genes. That is the ORF, which is coding for the protein, the structural gene. Then you have that codes the protein. Then you have the regulatory genes. They encode, they make products that interact with other sequence and affect the transcription and translation of these sequences. There are several of them. It can not only be protein, it can even be RNA also. Some RNAs are there, regulatory RNAs are also present. Then you have regulatory elements, heat stress elements, a particular sequence of DNA on which a particular transcription factor binds. So the control of gene expression, it is often it is controlled by controlling the initiation of transcription. When the transcription starts, it is controlled. And you have regulatory protein. They bind to DNA, either block or stimulate transcription depending on how they interact RNA polymerase. Just RNA polymerase is not causing the transcription. There are several transcription associated factors which are binding. Sigma factor, all these are binding. They are also having the Tata box below, to which the RNA polymerase is binding. They also have role. Some Tata boxes hold the RNA polymerase much weak and some much strong. Next slide, control of gene expression. So this gene regulation in prokaryotes, it's extremely is extremely high. In prokaryotes, it is extremely tightly regulated because bacteria live in an ever-changing environment. A bacteria, say E. coli, suddenly all glucose is removed from the media and you put them in the lactose medium. The genes responsible for metabolizing lactose is suddenly gets on, is switched on. Lactoperon. पढ़े हो ना lactoperon? And eukaryotic cells, they regulate gene expression to maintain homeostasis in the organism, balance in the organism. Next slide. Regulatory proteins. So gene expression is often controlled by regulatory proteins binding to specific DNA sequences. The regulatory proteins gain access to the bases of DNA at the major groove. So you know what major groove, minor groove is? DNA helix. One side is much more larger and one side is smaller. Larger side is major groove. So they have actually these regulatory proteins, say for example, heat shock transcription factor, it binds to heat stress elements when there is heat stress in the organism. So this heat stress binds to the major groove. So the elements are present in the major groove. It has been seen that if you change the phase, say from the major groove, you send the DNA sequence into the minor groove, the heat stress elements, suddenly the heat stress, it is not so good. And regulatory proteins, they possess DNA binding motifs. Okay, go to the next slide, regulatory proteins. Now next one, DNA binding proteins. You have to understand what is a domain and what is a motif. Regulatory domain in a protein, transcription factor, regulatory domain. You have a DNA binding domain, roughly 90 amino acids, 90 to 120 amino acids long. But that DNA binding domain is the whole region. If you remove some region, the binding is inefficient. But the point at which the DNA is binding to the protein, in the protein, that is the motif. Domain bada, motif chota. This classroom is my domain. यहाँ पे मैं जो भी बोलूं वो करना पड़ेगा सबको. It is the domain. But my motive is to sit and give a lecture over here. This small space. Classroom is my domain. Actual work I'm doing from here. वो क्लासरूम का एक या वो पंखा निकाल दोगे, I'll be in a discomfort teaching. ठीक से नहीं होगा. स्टूडेंट निकाल दोगे, जो होता है मैं मोस्टली स्टूडेंट्स आर नॉट देयर, क्लास भी नहीं होता है. But that is the domain. Everybody is part of the domain to make this teaching learning process. Similarly, in a protein, this is something. Next slide. So these domains are two types. First is the helix turn helix. Helix, alpha helix. Then there, you can see then there is a turn. Then another alpha helix. This is a transcription factor. You have a helix, that turn, another helix. So see how it is binding to the DNA. On the left side, A, helix turn helix. देखो DNA के ऊपर कैसे बाइंड कर रहा है. Helix turn helix. Then you have zinc fingers. Then you have leucine zippers. It's like a zipper-like configuration. Leucine zipper जैसे ये खुल जाता है inactive, लग जाता है active. Zinc fingers. So you have to practice these diagrams. Go to the आ सकता है. Zinc finger, leucine zipper. Okay, there are common DNA binding motif वाला slide में जाओ. This table is important. कहाँ bind करता है? Where it binds. Location, characteristics, binding site in DNA. This motif is in the protein, in the transcription factor protein. Domain is also in the protein. And on the DNA, you call them element, heat stress element, data binding element. Now we come to operon. ये tryptophan operon है. You have studied tryptophan operon, you have studied lac operon. When lactose is in the media, there is the 3 genes for lactose metabolism are transcribed. When lactose is not in the media, it is shut down. You have a sequence known as promoter operator. So tryptophan is positive operon, positive control, and lac is negative control. So tryptophan means, so when tryptophan is added in the media, then nothing happens. Okay. Tryptophan media में दे दिए, so you don't, the cell doesn't need to synthesize tryptophan. When tryptophan is not in the media, it is synthesized because the cell needs tryptophan. And negative ulta hota hai. Lactose. When lactose is there in the media, only then you need enzymes which is responsible for lactose metabolism. That is, when lactose is in the media, it goes and it binds to a place and it allows transcription. So these are the typical type of transcription initiation. So let us go to the lac operon. So go to the scene, go to this page where the lac control system, the diagram, this diagram. ये वाला diagram. You have promoter for I gene. Then you have the I gene. I gene, what it does? It secretes a repressor protein. Stops repressor. बंद कर देता है transcription of lactose genes. ठीक है? After that, you have a cap binding site, catabolite activator protein. After that, you have a promoter of lac operon. Then you have the operator sequence. Then you have the gene for beta galactosidase, gene for permease, genes for transacetylase. So Z, Y, A, they are the coding region. And the regulatory region, you see the marking from PI to O. That is the regulatory region. And the whole thing from PI to A, that is the lac control system. This is the structure of the lac operator. So go to this picture. So when lactose is not in the media, there is this lac repressor gene on the left side. It is causing the mRNA, which is producing the lac repressor polypeptide. What it is doing, it is going and it is binding to the cap binding site. Result is the lac repressor which is formed. देख रहे हो lac peptide and pink colored lac repressor. No lactose is present. It is coming and it is binding to the operator. And before the operator is the promoter for the lac gene. And that region as RNA polymerase has bound. But बीच में एक रुकावट आ गया. This repressor protein आ गया बीच में. धक्का खाके it's not going ahead. Stop. Clear? ठीक है तो? ना नहीं
Yes, sir.
clear? Okay, I'll tell it once more. Lac repressor, when there is no lactose, what happens? Lac repressor is making the repressor protein. Repressor is binding the operator. Operator is after the promoter site. So the RNA polymerase which binds to the promoter, it is hitting a target. That is the repressor protein. रुकावट है, दीवार है, I cannot go through the wall. That wall needs to be removed. Now, when there is lactose in the media, repressor protein is formed, but lactose has got an isomer known as allolactose. Changes into allolactose. A little part of it changes into allolactose. It's an isomer. Same molecular formula, but different structure. That is allolactose. Now what it does, it binds to the repressor protein. And it binds and the repressor, that's an allosteric configuration which takes place. And this repressor that cannot bind now to the operator. And it is free, RNA polymerase is free to keep on transcribing. Clear? Now we go to this picture. Glucose low, inducer present, promoter activated. So glucose is low. Inducer present. What is inducer? Lactose. And promoter is activated. Promoter काम कर रहा है. मतलब normally glucose कम है. So when there is glucose, lactose का कोई काम नहीं है. Lactose को बंद कर देता है glucose. We will see why. Glucose lactose को बंद कर देता है. Only when there is no glucose. But here glucose कम है. Lactose है. Inducer, lactose is present and the promoter is activated. So what happens is there is this cap binder, cap protein, which binds to cyclic AMP. Okay. So increase cyclic AMP. What happens? Glucose level is low. Cyclic AMP is high. जब glucose low होता है, cyclic AMP is high. Low glucose, high cyclic AMP. Cyclic AMP, it activates CAP by causing a conformation change. So when CAMP binds to CAP, CAP-CAMP complex, this comes and binds to this promoter. It induces more expression of the genes. Okay? Clear? Online students, is it clear?
Yes, sir. Yes, sir.
Yes, sir.
जब ग्लूकोज हाई होता है साइक्लिक एमपी को कम कर देता है ग्लूकोज व्हेन ग्लूकोज इज हाई so the cap complex cannot bind, no lactose, inducer is absent, repressor bind करे. So cap camp complex नहीं है, camp नहीं है तो cap कोई काम नहीं कर सकता है. It cannot induce expression and repressor is binding. Here, so this is the lactose operon. Now, let us go to eukaryotic regulation. बहुत सारे slides के बाद. We'll go to this picture of RNA polymerase. You can see with the RNA polymerase, there are several other factors which are working. The transcription associated factors or TAFs, factor B, F, E, H, TF2D. So there are many of them binding together to the core promoter region. So you have certain sequences, general transcription factors bind to the next slide, bind to the promoter region of the gene. Okay? RNA polymerase 2 then binds to the promoter to begin transcription at the start of start site. So the first nucleotide which is transcribed, that is +1. कोई जीरो नहीं रहता है, +1 के आगे -1 रहता है. Clear? There is no zero. +1 starts transcription. And then you have the sequences known as enhancers. They are DNA sequence to which specific transcription factors or activator bind, like heat stress transcription factor binding to heat stress elements. That heat stress elements are in the enhancer region. Go to the next slide. You see the activator is binding to the enhancer. Transcription factor, RNA polymerase, and all binding to the Tata box and promoter. Once everybody binds, enhancer loops the DNA. उसको लूप कर देता है. देखो, it's looping. नीचे देखो एक loop form किया, and it touches the RNA polymerase, and huge amount of transcription takes place. Such a complex is known as enhanceosome. लिख लो, enhanceosome. Enhanceosome. EN, enhance, EOSOME. Apart from
Sir,
this, there are other.
Sir, আরেকবার repeat করবেন please. Enhance এর আগে কি বললেন?
Enhanceosome.
ঠিক আছে স্যার, thank you.
Co-activators and mediators. They also help in enhancing the transcription. They are part of the transcription apparatus. So next slide, this is the enhanceosome. Activators, co-activators, general factors, they all cause transcription. Okay, so this is essentially regulation of gene expression, and we have talked only scratch the surface, that is transcription initiation. Next day we will go ahead with the next part in my syllabus. Okay? Any questions? Do you have any questions? No questions.